Aloha and welcome to General Pharmacology. We'll move to the PowerPoint slides and get started with the Section 2 material. Section 2 material is going to cover cardiovascular medicine. We're going to start with the basic fundamentals of neurotransmission. And then when Section 3 starts, 3.1 will start with central nervous system pharmacology. And when we get to central nervous system pharmacology, I want you to remember that this lecture would start Section 3 as well. And so this lecture is so nice, you can even watch it twice. And so this would be like the beginning of central nervous system pharmacology as well. So we're going to start with neurotransmission, and instead of taking a right toward central nervous system pharmacology, we're going to take a left, go to autonomic pharmacology and things that involve body regulation and eventually cardiac medicine. And so we start here with neurotransmission. By the way, neurotransmission was my favorite subject in college. That was my favorite thing to study, neuroscience and neurotransmission. And it was a lot of fun to think about. So some of the concepts that we'll talk about are very complex, but once you guys do this a few times, you'll get the hang of it. All right, we'll start really simple. The neuron is the basic cell of the nervous system. So the nervous system is made up of a collection of neurons. Neurons provide an instantaneous method of cellular communication, near instantly. Cells can communicate in other ways. When we talk about endocrinology, we'll talk about cells that will release chemical signals into the blood to communicate with other cells in the body, like the pancreas when it secretes insulin during high glucose levels. That communicates to the other cells to bring the insulin in. But neurons don't communicate in that manner. They communicate through an instant method of communication, and we're going to call that action potential. So again, here is somebody's cartoon of a neuron. There are all sorts of shapes and sizes. The longest neurons start in your motor cortex and go down to your spinal gray. Very short neurons are even less than a millimeter. They're called interneurons, teeny, teeny, tiny neurons. So there are unimaginable variations in neuron structure and functions. Again, the largest neurons are a few feet long, and the smallest neurons are less than a millimeter. And so if you study the shapes of neurons, you can see the cell body here is part of the decision-making process. All these inputs from other neurons come into here, and then it makes a decision, and then it sends the action potential down the axon. Well, here, the cell body's not involved in the inputs. The cell body's part of the axon here. And then in some neurons, this is like a sensory neuron, where we don't want the cell body to be involved in any kind of decision-making process. Whatever's going on down here, we want to relay that information up here with no decision-making process coming from this one right here. So there's all sorts of shapes and sizes of neurons, and there's a strategy involved in their decision-making depending on where their cell bodies are placed. And if you spend enough time reading about that, there are Kandel and Schwartz books out there that spend hundreds of pages talking about this stuff, and that is all very fascinating, at least for me. Purkinje fiber from the cerebellum, I believe. What your cerebellum does, it's in the back of your brain. It's called the little brain in Latin. But what it does is it listens to what the brain is telling the muscles, and then it senses what's actually happening, and then it readjusts to make sure that the muscles are actually doing what the motor cortex is ordering them to do. And this requires a lot of inputs, a lot of decision making before it gets to the cell body and then sends the signal back. And so this is very complex neuron. You'll hear about extrapyramidal symptoms, which is commonly used terminology, and that's called extrapyramidal because the pyramidal system is your voluntary motor movement system. And so when you tell your muscles to move and they move, that's your pyramidal system. Other movements that we'll talk about when we talk about Parkinson's disease, they're motions, motor movements that people cannot control, and they're outside of this voluntary control system. We call those extrapyramidal, and they get their name from this little pyramid-shaped neuron in your motor cortex. And so these pyramid-shaped neuron bodies are the cell bodies that make up the upper motor neurons, and they have a pyramid shape under the microscope like plenty of other neurons. But back in the day, they saw that pyramid shape and they said, oh, we're going to call the voluntary motor system the pyramidal system, and that's from this pyramidal shaped neuron cell body right there. Who knew? All right, but rather than spend an unimaginable amount of time talking about the unimaginable variations, we're going to just talk about some basic common features among neurons. All right, so again, here's my generic cartoon, someone's generic cartoon of a neuron. And then again, there's the cell body. We talked a little bit about that. See the nucleus in the center, that's shadowed in. That cell body is going to do all exactly the same stuff that we talked about in cellular physiology and more. So you can go back and look at 1.2 if you want to know more about what the cell body does. Today we're going to talk about this electrical signal called an action potential. So the tube that it's going down is called the axon, and it's wrapped in these little myelin sheaths with these little in-between nodes, saltatory conduction. We won't talk about any of that today. However, that little electrical signal is supposed to show you that this action potential, that little lightning bolt going down the axon, is supposed to show you that the action potential starts in one place and goes the other. And they only go in one direction down an axon. So an action potential is the electrical signal that's carried down the axon, away from the sensory side and towards the output side. And so in our generic, it's from the cell body to the synaptic terminals. And then action potentials are only carried one way. If you read big, thick Candel books, they'll talk about interneurons that do have the ability, teeny tiny interneurons have the ability to do this two ways. They're super rare, at least in clinical pharmacology, so we're not even going to talk about that. And so this is correct right here. When we talk about neurons and action potentials of pathways that can be named in the brain and out to the body and vice versa, those action potentials are only carried in one direction. Do not confuse the term nerve with neuron. A nerve is quite simply a collection of neuron axon fibers, and it will describe these nerves and the signals in the axon fibers by the direction that they go. And so an axon can carry a signal away from the central nervous system, out from the central nervous system, and we'll call that efferent. And then signals can come into the central nervous system, and we'll call those afferents. And so we'll get a little philosophical on you. The central nervous system has efferents, and those are outputs. And technically, the outputs run from your body to muscles. Some of them you can consider smooth muscle, but truly your efferents are your striated muscle movement. Well, if it's going into your central nervous system, then it must be sensory. And so afferents are sensory, and you have all sorts of sensations. And efferents, your outputs are exclusively moving muscle. And my question to you as a human, an enlightened human, is the only output that comes from your brain really about moving muscles? Because when we think about things in neurology, our choices are this: efferents, outputs, afferents, input. And I would ask you, as a human, what are your efferents? Are they just quite simply striated muscle locomotion or some smooth muscle stuff that you have very little control of? What are your outputs as a human? Back to our regularly scheduled program, I used to have a really nice slide of this. I try to find what I'm looking for in my slides, and they're not here today. And so you Google neuron axon fiber, a slice through it, an electron micrograph, it'll look like this. You'll see that these are little, as if you can see me pointing, these are axon fibers, and the collection of them make up the nerve. Now, nerves can be efferent, meaning they're carrying axon fibers that exclusively go out. Nerves can be afferent, meaning they're carrying sensory information on the way in. And some nerves can carry axons. Some of them are motor axons going out. Some of them are sensory inputs coming in, afferents in, efferents out. And so nerves can be afferent, efferent, or both. However, the axon fiber itself can either be one or the other. All right, so here our action potential is coming from the hillock of the cell body and going down to the synaptic terminals. And this action potential is causing a chemical signal to be released from the synaptic terminals, and we're showing you that as these little green dots. And so as my electrical signal comes down the axon, by the time it gets to the synaptic terminals, it causes a chemical signal to be released. And so let's take a closer look at this chemical signal where this neurotransmitter is being released. So the neurotransmitter is the chemical signal coming from the synaptic terminals, and the synaptic terminals are releasing the neurotransmitter because of action potential. All right, so here we're at the end of the neuron, one of the synaptic terminals, and we can see these little blue packets of neurotransmitter, and they get released. The neurotransmitter gets released into this synapse, and then the blue part, the vesicle, that phospholipid bilayer, automatically joins with the cell membrane. It's fun to talk about. This is what I want you to know. The neurotransmitters, the green dots, stored in packets called vesicles, and those are the blue circles. And again, when the vesicle releases its neurotransmitter, that vesicle ends up being part of the membrane. And it's the action potential that causes this to be released, and so I show that with the flashing neuron being the action potential. All right, so what I want to show you is that we're going to look at this picture over and over again. We're going to give it different names. Sometimes we're going to call it the sympathetic nervous system or the parasympathetic nervous system or the neuromuscular junction. And you're going to notice that the picture stays exactly the same. Action potential causes neurotransmitter to be released, and then it'll be sensed on the other side by something. And so that's what we're going through. All right, so the neurotransmitter is released into a space, and that space is called the synapse. And so you'll hear presynaptic this and postsynaptic that, and all sorts of synaptic terminology, like you guys are neuroscience experts. And that's really quite simply the synaptic space right there, the synapse. And that's where the neurotransmitter is released into a space. And so neurons can communicate with muscles and organs. So when the neuron communicates with the muscle, we'll call that the neuromuscular junction. During autonomic pharmacology, we'll talk about neurons affecting the organs. And then when we get to central nervous system, that'll be about neurons that interact with other neurons. And regardless, that space is called the synapse. All right, so now we know which is the presynaptic membrane. Well, the vesicles carrying neurotransmitter are released from the presynaptic membrane, and then that neurotransmitter is sensed either by a muscle or an organ or another neuron, depending on where it is, by this postsynaptic membrane. All right, so on this postsynaptic membrane are receptors that sense the neurotransmitter. If you remember the lock and key lecture that we gave in 1.3, here these little keys are being released from this neuron, and those little keys are finding the place where they fit perfectly in the postsynaptic membrane. All these in a synapse, is only one neurotransmitter being used in that synapse, and we'll talk about which neurotransmitters are used in which type of synapse. And then all these receptors are there that are specific for this neurotransmitter. So neurotransmitters are chemical signals released from neurons to communicate with muscle, organs, and other neurons. The neurotransmitters that we'll talk about in this course are acetylcholine or acetylcholine, whoever's arguing with me that day. Catecholamines, dopamine, norepinephrine, and epinephrine. Dopamine, norepinephrine, and epinephrine are referred to as the catecholamines. Please do not confuse this col word with that col word up there, acetylcholine, catecholamines. They are not related to each other. The catecholamines are dopamine, norepinephrine, and epinephrine. You take dopamine and you manufacture norepinephrine with the dopamine, and then you manufacture epinephrine with the norepinephrine. And so each is the precursor for the next. And so dopamine, norepinephrine, and epinephrine are the catecholamines. Serotonin is 5-hydroxytryptamine, 5-HT. And what's confusing is that when we talk about antidepressants and disorders of mood, we'll talk about norepinephrine and serotonin as if they are two neurotransmitters that are related in mood disorders, because they are. And so we'll think of them as cousins, and sometimes we'll call them the monoamines because they're very similar to each other, especially in effect with disorders of mood. But serotonin, despite being a monoamine, is not a catecholamine. All right, so the catecholamines are dopamine, norepinephrine, and epinephrine. That's something that you're going to want to know if we're going to use neurotransmitter words in a pharmacology context. All right, well, you've heard of antihistamines. Histamine is a naturally occurring substance in the body that mediates allergic reactions, but something you'll see is that histamine is also a neurotransmitter. And you'll notice that if you dig deeply into neuroscience material, there's this thing called biologically active amines. Any of the amino acids, if they're shaped a certain way, can have effect on receptors. And we can come back to that eventually. Gamma-aminobutyric acid is GABA, and it is made from and then recycled into glutamate, and we'll talk about glutamate and as a neurotransmitter as well. And so this is the list of neurotransmitters that we will talk about in this course. But we're going to start with acetylcholine. Now, we talked about acetylcholine a little bit in 1.2 when we were talking about how a protease enzyme works, and so we talked about acetylcholinesterase breaking down acetylcholine into its parts, and so acetate and choline. And so to make acetylcholine, we start with acetyl coenzyme A. This is made by the mitochondria as part of the Krebs cycle system, and then choline comes from lecithin, which is a fatty acid. And your body takes these two things and makes acetylcholine. When we get to the section 3 material, we will talk about acetylcholine in the central nervous system. That's for 3.1 neurotransmission. Before we get into autonomic pharmacology and how acetylcholine affects the organs, you need to know that acetylcholine is also in the neuromuscular junction. And that's why if you study acetylcholine drugs very carefully, they can have an effect on the central nervous system as well as the neuromuscular junction. And then we'll talk about this as acetylcholine affecting the parasympathetic nervous system. Things like heart rate and lungs are affected by acetylcholine as well. So here we are at the neuromuscular junction. The neuromuscular junction carries the neuromuscular junction is the synapse between the lower motor neuron and the muscle, the striated muscle. And that receptor type is called nicotinic. And that's because nicotine sticks to it in a petri dish. When we came up with, when they name these things, they figure out what kind of chemical signal has the greatest affinity to it. So they call it a nicotinic receptor because it has a high affinity for nicotine. Now, when people use nicotine as a drug, that effect comes from the central nervous system, nicotine's effect on the central nervous system, not the neuromuscular junction. And so be careful using that word nicotine because the nicotinic receptor in the neuromuscular junction just has a high affinity for the chemical nicotine. All right, so here we are at the neuromuscular junction, and I've taken my same neuron cartoon and my same postsynaptic cartoon. And here we have a lower motor neuron that starts in the ventral gray matter of the spinal cord, and then it goes out to voluntary muscle. And so acetylcholine will always be the neurotransmitter used in the neuromuscular junction, and it's going to be sensed by nicotinic receptors. All right, so myasthenia gravis is very rare. People ask me why I talk about this. I'm like, well, sometimes disease processes, many times disease processes, every time disease processes teach us something about physiology. And so here, the acetylcholine is being released to signal the voluntary muscles to move. So what do you think happens if those receptors are blocked? And here in myasthenia gravis, the body makes antibodies that block those receptors in the neuromuscular junction. And so what do you think happens? Those little Y-shaped proteins are my antibodies made by the immune system, and they're supposed to be out looking for foreign substances like COVID viruses and monkeypox, but they've made a mistake in this case, and they've decided to stick to nicotinic receptors in the neuromuscular junction. And so what do you think happens? Well, it causes weakness, and this weakness is caused by antibodies that block acetylcholine receptors in the postsynaptic neuromuscular junction, inhibiting the effect of the neurotransmitter acetylcholine. And so I cut and paste this out of some explanation, and so now hopefully this makes more sense now that we've looked at the cartoons. So here is the problem right here. Drugs that affect acetylcholine are going to be used to diagnose and treat myasthenia gravis. Those same drugs are going to be used for all sorts of other reasons as well. And so that's why we're going through this, because you'll think, oh, well, that's a myasthenia gravis drug. And they're like, well, they use it over here and for all sorts of other reasons. And we're going to try to explain why in this lecture and the next lecture. All right, well, we're playing Where's Waldo with acetylcholine. Where did we find it? In the neuromuscular junction. Where else are we going to find it? In the parasympathetic nervous system. And this is what we're going to talk about the next time. The autonomic nervous system is 2.2. And that's probably the most important lecture in the entire series. So this and autonomic pharmacology, something you want to have a really good handle on, because that's really the most complex pharmacology that there is. All right, so the autonomic nervous system, it signals smooth muscles in the organs, and that uses acetylcholine. The muscarinic type is called, the receptor type is called muscarinic because of muscarine toxin. I think that's from mushrooms, has a high affinity for these receptors, muscarinic receptors. And so I bring this up because if we talk about a muscarinic drug, we're talking about a drug that affects the parasympathetic nervous system. We're not talking about the neuromuscular junction. All right, so muscarinic receptors are not going, muscarinic drugs are not going to affect nicotinic receptors and vice versa, and that's why we talk about that. All right, so again, the autonomic nervous system uses acetylcholine to signal parasympathetic smooth muscle. And again, those receptor types are muscarinic. And so here is my cartoon again, and all I've done is just change the labels, even though these are different neurons in different places. And so here's a parasympathetic neuron. You've heard of the parasympathetic nervous system if you took classes that talked about the vagus nerve. The vagus nerve affects the lungs, the vagus nerve affects the bowels, the vagus nerve signals the heart as well. And so the vagus nerve carries the largest parasympathetic organs, largest parasympathetic pathways coming from the cranial nerves. However, there's other parasympathetics going to your pupils, going to your lacrimal and salivary glands as well. And all of them, all of the parasympathetic neurons that all of the neurons that end up terminating on an organ will release acetylcholine, and those receptors will be called muscarinic. When we talk about terminating on an end organ, we'll use a word peripheral, meaning outside. We'll talk about central neurons. That's in the central nervous system. When we talk about the peripheral synapses, we're talking about the neuron affecting the end organ here. All right, well, the antibodies, rather than finding monkeypox and COVID, maybe the antibodies will stick to the lacrimal glands. They make tears, and the salivary glands, they make saliva in your mouth. And we can have an autoimmune disorder release antibodies that stick to lacrimal and salivary glands. And so Sjogren syndrome is something interesting to read about. Those antibodies will block the muscarinic receptors on the salivary glands and lacrimal glands. It gets far more complicated than this, but this is a great place to start. So Sjogren syndrome causes dry eye and dry mouth, quite simply from the circulating antibodies that block acetylcholine receptors at the muscarinic receptors on lacrimal and salivary glands, therefore inhibiting the effect of the neurotransmitter acetylcholine. So not only are drugs that mimic the effects of acetylcholine used to diagnose and treat myasthenia gravis, a disorder of the neuromuscular junction, drugs that mimic the effects of acetylcholine are used to diagnose and treat myasthenia gravis are also used to treat Sjogren syndrome as well. And so that's why the acetylcholine business gets really complicated in pharmacology land. And that's why we're going to go through this very carefully. Hopefully you have a very good handle on this before we move forward. All right, so drugs that mimic the effects of acetylcholine will be called cholinomimetic. And so if you come across that word, that's just drug that mimics the effects of acetylcholine. And drugs that mimic the effects of acetylcholine, cholinomimetic drugs, are used in the treatment of bowel problems, bladder problems, the eye disease glaucoma, myasthenia gravis, Sjogren syndrome, anticholinergic toxicity. If you're in the anesthesia business, the surgical anesthesia business, you will be reversing certain paralyzing agents with cholinomimetic drugs. And then we'll come back in central nervous system pharmacology and talk about cholinomimetic drugs in the treatment of Parkinson's disease and Alzheimer's disease. And so the next lecture is really about, sure, this cholinomimetic drug is designed for Parkinson's disease, but it might have an effect on bowel and bladder and the organs of the autonomic nervous system. And so when we talk about the autonomic nervous system, one of the things we'll say is drugs that affect any one of the organs in the autonomic nervous system will affect all of the organs. And so if you look at the labels of unwanted effects of any of these cholinomimetic drugs, they will include all sorts of things that have to do with the autonomic pharmacology. All right, so where did we find acetylcholine? We found it in the neuromuscular junction, and we brought that up because we're going to spend a lot of time talking about acetylcholine in the parasympathetic nervous system, and that is what the next lecture, 2.2, Autonomic Pharmacology, is about. But we bring up acetylcholine in the neuromuscular junction because some of the classes of drugs we use in myasthenia gravis will be the same classes of drugs that we're going to use for all sorts of bowel and bladder and autonomic organ pharmacology. And so that's why we bring that up. All right, to get through autonomic pharmacology, the next lecture, all we have to know about the catecholamines is actually norepinephrine and epinephrine. We'll talk a lot about dopamine and the disease processes related to dopamine when we get to central nervous system pharmacology. However, for autonomic pharmacology, all you really need to know is acetylcholine, neuromuscular junction, nicotinic receptors. Acetylcholine, parasympathetic organ synapse using muscarinic receptors. For the catecholamines, all we'll talk about in autonomic pharmacology is norepinephrine and epinephrine. We'll talk a little bit about dopamine. All right, catecholamines are made from the amino acid tyrosine. I hope we go through that at the end of the lecture. We'll talk about all the catecholamines. We don't really talk about epinephrine so much when we talk about the central nervous system, but dopamine and norepinephrine are things that we'll talk about in the central nervous system. But the autonomic nervous system is where we're going to talk about the catecholamines next. However, I'll tell you this now, the neurotransmitter that signals the sympathetic smooth muscle is epinephrine and norepinephrine, and so we'll talk about the neuron producing mostly norepinephrine. We'll talk about the adrenal gland making mostly epinephrine. We'll try to make that really simple, and then basically in this course we're just only going to have two receptors, alpha and beta. There's dopamine receptors, and there's alpha 1 and alpha 2 and beta 1 and beta 2, and it gets more complicated than that, but we're going to start right here with alpha and beta being the receptors for epinephrine and norepinephrine. When we get into central nervous system pharmacology, we're going to see that dopamine and norepinephrine seem to be completely unrelated when it comes to its function in the central nervous system. Something I can tell you right now, a fun thing to know about autonomic nervous system is whatever epinephrine does in the autonomic nervous system, norepinephrine is going to do about the same thing. It's going to have the same effect. It's going to have exactly the same effect, just not as efficacious. And so some receptors are much more sensitive to epinephrine than norepinephrine. Other receptors are more sensitive to norepinephrine than epinephrine. If you read carefully, you'll come across that. But I'll tell you right now, as an effect on the autonomic nervous system, their effects are going to be the same. The receptor types are alpha and beta. Same cartoon, different labels. Your sympathetic chain ganglia come from, your sympathetic chain comes from your cervicals as well as your sacrals, and we'll talk mostly about the cervical ones. And they send a neuron to your vascular smooth muscle in this case. And so your vascular smooth muscle, they go around arteries and arterioles. When you're laying down and you stand up, your blood would go straight downhill to your legs unless you had a system to squeeze those blood vessels and push the blood upwards. And so when you go from laying down to standing up, you need those blood vessels to squeeze to push blood uphill. And so around the blood vessels are these smooth muscle that squeeze, making the artery more narrow, pushing blood upwards, raising pressure. And that smooth muscle is mediated by a sympathetic neuron that releases norepinephrine into the synapse, and it's alpha receptors that sense the norepinephrine, sense the catecholamine, and respond by tightening the muscles around the blood vessel, raising pressure. So then it makes sense later on if we give somebody an alpha blocker that will do in certain cases of prostate hypertrophy, benign prostatic hypertrophy. People will take alpha blockers for that. People will take alpha blockers for high blood pressure. But again, what are we doing? We're blocking the system that allows vascular smooth muscle to respond from going from laying to sitting to standing. And so when people are taking alpha blockers, we have to be really careful about them not having orthostatic hypotension, meaning when they go from one position to an upright position, the body can't compensate and push blood up into the brain, and they can get pre-syncopal, lightheaded, and even pass out, be syncopal. So we have to be really careful about people who take alpha blockers when they're changing position. All right, well, if we're talking about the heart or lung smooth muscle, we'll talk about beta receptors. And again, the neurotransmitter is being released into the synapse is norepinephrine. So for 2.2 autonomic pharmacology, this is all the neurotransmitters you will need to know. Acetylcholine, parasympathetic nervous system. Catecholamine, sympathetic nervous system. All right, so after 34 minutes, the next lecture really just comes down to this right here, acetylcholine and the catecholamines. All right, let's talk a little bit more about neuron membrane physiology and some important concepts in clinical pharmacology. And so we'll start with this. Here is a question. Here, the postsynaptic membrane has been excited by the neurotransmitter. Well, what makes it stop? Maybe this is the neuromuscular junction. Maybe you're moving at the mouth or trying to do something fine-tuning with your hands, and so the signal says, okay, move. Well, then how do you make it stop? How do you have this precise on-off effect at the postsynaptic membrane? And there's basically two ways to do this. There's either reuptake pumps or breakdown enzymes. And so here is a close-up of my synapses. Here the neurotransmitter is signaling the postsynaptic membrane, and somehow that has to stop. Well, one of the ways we can do this is with a reuptake pump. And another way we do this is with breakdown enzymes. And we talked about this with acetylcholinesterase. Acetylcholine goes into the neuromuscular junction, and what makes it stop? Well, the little breakdown enzymes break it down immediately. So when the acetylcholine's there, we move, and then the enzymes break it down immediately. So again, we talked about acetylcholinesterase, the enzyme that breaks down acetylcholine, and we talked about this in 1.2 cell-phys. So acetylcholinesterase breaks down acetylcholine in the synapse. So if we block acetylcholinesterase, we will end up with more acetylcholine in the synapse. And so this is what confuses everybody. This is why we talk about this over and over again. So acetylcholinesterase inhibitors in pharmacology land, we call these cholinesterase inhibitors, and cholinesterase inhibitors are cholinomimetic, are indirectly cholinomimetic, mimicking the effects of acetylcholine. Why? Because we're blocking the garbage disposal that breaks down the acetylcholine, leaving more acetylcholine behind. And so when you see that word cholinesterase inhibitors, remember the inhibitor is stopping the enzyme that breaks down acetylcholine. That's why acetylcholinesterase inhibitors are cholinomimetic or cholinergic, and they do that indirectly. So again, if I block acetylcholinesterase with cholinesterase inhibitors, I leave more acetylcholine as if I used a drug that is a cholinergic agonist. And I did that indirectly by blocking its breakdown, and we'll see this strategy in pharmacology throughout. So these cholinesterase inhibitors will be used in the treatment of myasthenia gravis or Sjogren's syndrome or bowel or bladder problems, glaucoma, anticholinergic toxicity, belladonna toxicity. We'll use these as reversing agents and in anesthesia. As well as Alzheimer's and Parkinson's disease. And that's what makes this confusing for people, because they'll see a drug and they're like, oh, it's used in glaucoma. Well, that's okay. It's also, that same drug can be used for any of these issues as well. Some typical cholinesterase inhibitors are physostigmine, neostigmine, pyrrotostigmine. The stigmines are cholinesterase inhibitors. They are cholinomimetic by blocking the breakdown of acetylcholine. And they have all sorts of interesting uses. So be careful when we talk about the stigmines, because they have all sorts of cholinergic effects on all sorts of organs of the autonomic nervous system. Edrophonium tensilon is a popular cholinesterase inhibitor. It is used to diagnose and treat myasthenia gravis. And so one of your questions in your clinical discussions is, well, what is the tensilon test? And how is it used to diagnose myasthenia gravis? Other cholinesterase inhibitors we'll use in dementia for Alzheimer's and Parkinson's disease. These are cholinesterase inhibitors as well. So the cholinesterase inhibitors, the point is to not know exactly what the cholinesterase is used for. What I want you to see is that all of these drugs, if you look at the label, have something in common. They all have the effects of a cholinomimetic drug. And when we talk about we'll talk about the effects of the cholinomimetic drugs on the autonomic nervous system next time. Insecticides are cholinesterase inhibitors as well. Insects rely on their acetylcholine systems and are very sensitive to cholinesterase inhibitors blocking the breakdown of acetylcholine. Military nerve gas is also a cholinesterase inhibitor, an irreversible cholinesterase inhibitor as it's described. And so if you know somebody in the military, they'll carry an atropine injector. It will also have a cholinesterase reactivator in case they come in contact with nerve gas in a combat setting. And so we'll talk about atropine being an anticholinergic drug because remember these are cholinergic drugs increasing levels of acetylcholine. These are acetylcholine poisons that increase levels of acetylcholine, so we'll use an acetylcholine blocker like atropine in this treatment. We'll talk about the reuptake inhibitors. Remember, reuptake is how we get the neurotransmitter out of the synapse, and we'll talk about this when we get to central nervous system pharmacology. All right, let's talk a little bit more about receptors. Remember, when we first started talking about locks and keys, we said, well, it's not really the key that determines what happens, it's the lock that determines. If you put the key in the ignition, it starts the car. If you put it in the trunk, it opens the trunk. And so what the receptor does determines the effect, and we're going to divide these receptors into two types, gated ion channels and second messenger systems, ionotropic and metabotropic receptors. All right, so let's talk about gated ion channels. What I want you to know about them is they give us an instant on and off. And so when we're talking about the neuromuscular junction of voluntary muscle, we need instantaneous on and off communication. When the neurotransmitter goes in, we want it on. When it's broken down, we want it off. Neurotransmitter present, on. Neurotransmitter not present, off. And we want it to be instantaneous. And so this is what we'll use in the neuromuscular junction, as well as sensory neurons, other places. Oh, that didn't work properly. There we go. So there's my neurotransmitter coming in contact with the receptor, and it opens a channel that allows sodium to come into the cell. The sodium has a need to rush into the cell, not only from its electrical, but its concentration gradient. And so here again, we have a neurotransmitter that's coming in contact with the receptor and then opening a channel, causing a change of voltage across the membrane. So this gives us an instant on and off method of communication. We'll talk about the voltage-gated ones later. So if it's where is that? If it's ligand gated, that means a chemical signal opens it. If it's voltage gated, that means a voltage change opens it. But again, by opening that channel, we can cause an instant voltage change across the membrane. All right, we'll skip all that. We're gonna come back to that. All right, well then another way that receptors can work is by not just being an ion gate, but by a second messenger system, and this causes gradual metabolic changes to occur. So think about it. When your sympathetic or parasympathetic nervous system signals your heart, the sympathetic signals it to speed up using epinephrine or norepinephrine. Acetylcholine will make the heart slow down because that's what the parasympathetic system uses. And when we want the heart to speed up or slow down, we don't want this instant fast, slow, fast, slow. The heart doesn't do that. We tell it to speed up. You're panicking. Your heart starts to beat really fast. Well, it's going to keep doing that until that wears off. It takes a long time. And so some things we don't want instant on and off. We kind of want gradual metabolic changes to occur. When you signal your heart to beat really fast and you're panicking, your heart needs some metabolic support. There's all sorts of stuff that has to go on inside the heart to provide the nutritional needs that are required for this increased heart rate. And so when you see drugs that affect end organs, you're going to notice that they're going to use a second messenger system. So here my neurotransmitter is coming in contact with the receptor and then signals these chains of proteins to its end protein right there. Here's adenyl cyclase, and it's going to take ATP and make cyclic AMP. And cyclic AMP is one of the second messenger systems that you will see. Now remember, we had A and G and T, A, G, C. Anyway, you'll see any of the nucleotides used in the second messenger system. ATP is most common, GTP is less common. However, any of the nucleotides can be used in a second messenger system. ATP to cyclic AMP is most common, so cyclic AMP is the most common. Again, you'll see cyclic GMP if you read carefully. But they all basically work the same. Some kind of chemical signal outside of the cell activates the cyclic enzyme inside of the cell, causing an increase in cyclic nucleotide second messengers. And this increase of that chemical signal causes all sorts of changes to occur in the body with receptor regulation, metabolism regulation, DNA regulation, and it can activate or deactivate any of those receptors. And so it gets really complicated. And so again, if you think about it, when you speed up or if you slow down the heart rate or speed up the heart rate, if you speed the heart rate up you're increasing its metabolic demands. And so you don't want this instant on and off. You want this chemical signal that tells the internal parts of the heart muscles to change their metabolic activities to support what's going on. And so that's basically why we describe our receptors into the gated receptors that allow instant on and off, and then the second messenger systems that allow slower, gradual metabolic changes to support whatever's going on with that organ. All right, well, we talked about breakdown enzymes. Here, there's an enzyme that breaks down our second messenger, and so cyclic AMP is broken down by phosphodiesterase into cyclic 5' AMP. And again, you'll see cyclic GMP broken down into 5' GMP. This can be done with any of the nucleotides, but again, cyclic AMP is what's most common. But phosphodiesterase breaks down cyclic AMP. So one of the many things that caffeine does is inhibit phosphodiesterase, and we'll talk about other phosphodiesterase inhibitors in here as well. And one of the things that phosphodiesterase inhibitors do is raise cyclic AMP levels. All right, so when we first started, what raised cyclic AMP levels? Oh, a chemical signal outside of the cell eventually rose cyclic AMP levels. Well, here we're blocking an enzyme inside of the cell and still getting the result of raising cyclic AMP levels. And so that's what you'll see. That's why it's important to think about the different ways we can accomplish the same thing in pharmacology. I think we've made this point, and this point as well. Let's talk about action potential. Oh, I'm moving quickly today. Excellent. All right. To know about action potential, we have to talk about sodium, potassium, and chloride. And then to release vesicles, we need calcium. Calcium is required to activate the synapse to release neurotransmitter or release any of the vesicles. When we talk about white blood cells releasing their contents, calcium's involved. So calcium's very important in muscle movement and release of vesicles. As I go through action potential, we'll talk mostly about sodium and potassium. All right, you guys know what table salt is. It's sodium chloride. That's the white powder you put in the water, and it divides into its ions, sodium ions and chloride ions. Same thing with potassium chloride. And so these are the stars of our show. We'll start with sodium-potassium pump, otherwise known as sodium-potassium ATPase. And it's an enzyme that uses ATP for energy and converts that energy into pumping sodium out of the cell and pumping potassium into the cell. I did bother to make that 3-to-2 ratio that some people like to talk about, so we're pushing more positive charges out of the cell. At the same time, we're pushing positive charges inside of the cell. This helps make the outside of the cell more positive. Large proteins outside of the cell are positive as well. And so we create this voltage on the outside of the membrane. All right, so serum is an extracellular fluid. Remember, we're pumping it out of the cell and into the extracellular space, and we're doing the opposite with potassium. And so serum is an extracellular fluid. That's why sodium in the serum is 135. And potassium is really, really low. Why? Because potassium is intracellular. And so as people die and the ATP is not able to pump the sodium-potassium pumps, one thing that we see is potassium come way, way up because we're constantly trying to keep our potassium inside of our cells. And again, the system uses ATP to power the sodium-potassium pump. And what this does is create a sodium workforce. And so many times we talked about the sodium force driving something into the cell, and the sodium workforce was created by the sodium-potassium pump at the membrane. So there's more concentration of sodium outside than inside. That's what a concentration gradient is. And if you want to see how a concentration gradient works, take a big glass of water and put a drop of food coloring in it, and the food coloring starts when the drop goes in. That area is very high concentration, and the place where the dye is not is very low concentration, and you'll see that food coloring is going to move in every direction it can until it uniformly fills up that space, and that's called the concentration gradient, and your body knows how to put that to work. It's also more positive outside of the cell, and so this pushes positively charged sodium into the cell. So when we're talking about the sodium workforce, we're talking about both a concentration gradient and an electrostatic gradient. And so we'll call it the sodium force. That's my sodium ion with that green arrow showing you it's trying to vector its way inside of the cell. The sodium-potassium pump does not come on and off. There's all sorts of books and instructors that tell you that the sodium-potassium pump comes on and off to reset the membrane. That is not true. The sodium-potassium pump is always running to create this electrical workforce. What it's really doing is creating a battery. For those of you who know anything about physics, the sodium-potassium pump is turning ATP into a capacitor, essentially a battery. And so every point of the membrane can utilize that battery without waiting for ATP or needing ATP for energy. And so that's what's very interesting about the sodium-potassium pump. And then we use that electrical energy to do work. So the sodium force can be used to transport nutrients across a membrane, and we showed you this in the second lecture, this sodium force drives glucose as well as amino acids into the cell through facilitated transport. And we can use this force to do work as an action potential. Because the sodium force, the sodium gradient powers action potential, it makes sense that we'll talk about sodium channel blockers as anesthetics, because sodium channels are required for excitable membranes. Blocking them is going to interfere with their function. And so those of you in the dental hygiene business, we'll start right now, lidocaine and all of its esters and amide cousins are sodium channel blockers. Because sodium channels power action potential, it makes sense that sodium channel blockers would be used for all sorts of things to interfere with excitable membranes, including their use as local anesthetics. All right, so the action potential is a wave of voltage change that occurs by opening and closing of ion channels. And so the first is sodium rushing in, and then you'll see potassium rushing out to compensate. And so we'll start here. Here's my sodium-potassium pump, and it's pumping sodium out of the cell, potassium into the cell, and it's creating this battery across the membrane. It's creating this polarized electrical signal across electromotive force across the membrane. So it polarizes the membrane. That means the voltage on one side is different than the voltage on the other side. All right, so there's my neurotransmitter, and it has signaled a sodium channel to open. We'll call it a slow channel. All right. Well, now that I'm moving a positive charge across the membrane, right here in this area, there's a voltage-gated sodium channel, and it senses this voltage change because we're letting sodium across the membrane. That's going to cause a voltage change, and that's sensed by the sodium channel. And what does it do? Just allows sodium just come rushing in a fast channel. So now I have sodium rushing in. All right, well, what's going to happen when sodium comes rushing in? right here in this local area, right here on this small part of the membrane, well, it's not going to be polarized anymore because the positive charges are moving across so quickly. So moving all that sodium so quickly across the membrane, and that spot causes that spot to be depolarized. And so now this is electrically neutral because the positive charge on the outside has been compensated by all this sodium that's rushed in right here on this part of the membrane. Well, that's sensed by voltage-gated potassium channels. And they sense that depolarized membrane, and what do? Well, they open up and allow sodium to come rushing out. Why? Because it has a concentration gradient. Remember, there's more potassium inside of the cell than outside of the cell. And because in this area right here, it's depolarized, this creates a force that allows potassium to go outside of the cell through its potassium channel. Now I'm restoring the positive charge in this little local area right here, resetting the membrane. And so when we talk about excitable membranes, whether we're talking about the heart or the neurons, we'll talk about cardiac excitable membranes, the myocytes, when we talk about 2.6, and we'll see it's opening potassium channels that resets the membrane, repolarizes the membrane, and prepares it for another action potential. And this occurs over and over and over again in a wave. So this would be a membrane of an action potential. Here, this would be the membrane of an axon. And what I wanted you to see is the action potential moving down the axon. And what we're seeing with the blues, those are opening the sodium channels, and the sodium comes rushing in. And then you'll notice right in that local area, the red part is the potassium channels opening to recompensate the membrane. We have to give it time for it to working. And so look, we'll point at this part of the membrane right here. All right, it's polarized, it opens, depolarized, potassium compensates, now it's reset, sodium channels open, now potassium compensates, sodium channels, potassium channels. And this is always going on to create this wave of voltage change down the membrane. So I think all of this stuff is really incredible. This is basically what I devoted my undergraduate to. It took me a little time later to realize I was just really interested in everything, not just neurons. So take some time to let this stuff sink in, read as much as you can about this stuff, because it'll be very important. All right, in the short time that we have left, let's go over some things that you'll need to know. Not so much for the next lecture, but this is really the place that I fit it in because you'll need to know these things when we start talking about central nervous system pharmacology. So I'm going to start throwing the words at you now. You know what the word amino acid means. There's my amino group. There's my carboxylic acid moiety. And then the R is the variable group. Right? We know this, amino acid. And the variable side chain. Right? Great. So when we talk about the biosynthesis of amino acid-based neurotransmitters, we're not talking about acetylcholine. Acetylcholine is very unique, that its precursor comes from the mitochondrial Krebs cycle. But all the other neurotransmitters we'll talk about come from amino acids. Those are the catecholamines. These are the monoamines. Sometimes we call dopamine the monoamine because it is, but when we talk clinically about the monoamines in mood disorders, we're talking about norepinephrine and serotonin. All right, so remember, it's dopamine, norepinephrine, and epinephrine that are the catecholamines. It is serotonin and norepinephrine that are the monoamines, and dopamine technically is too. Serotonin is 5-hydroxytryptamine. We're looking at all these amine words. Gamma amino butyric acid, histamine. And so we talk a lot about biologically active amines for this reason right here, because these amines have all sorts of effects on the body, as well as being neurotransmitters. And so you'll see these mixed effects like histamine. When I was in college, I only wanted these things to do one thing and nothing else. And so histamine was cool when it was coming from white blood cells making me itch. But now I know that histamine is a neurotransmitter found in the stomach. And so that's where this word biologically active amine comes from, because these have different uses in different places. I wanted serotonin to be this great neurotransmitter in the central nervous system, and it is, but it got its name, blood-tonin. It's a vasoconstrictor released by platelets. And so there's mixed action of these as we go through them. And that's where biologically active amines comes from. All right, well, when you get lost somewhere around the synthesis of when you get lost in the synthesis of epinephrine, I just want you to know these two steps right here: hydroxylation and decarboxylation. All right, these are the two major steps in amino acid synthesis, and you can forget everything else as long as you just remember hydroxylation, decarboxylation. All right. So hydroxylation is quite simply the hydroxylase enzyme putting a hydroxide group on the R group. We saw this when we went from phenylalanine to tyrosine, and we talked about those being cytochrome enzymes. And if you read carefully about places where you will find catecholamines, they'll refer to these colorful enzymes. So here we're calling it a hydroxylase, and the real point here I'm trying to make is that the hydroxylase just puts a hydroxide on, usually the R group. And so this is a very important step in amino acid synthesis. Right? That's what the hydroxide group is, that oxygen group. All right, let's start over with my amino acid, and instead of hydroxylating it, let's decarboxylase it. All right, there's my carboxylic acid, and when we remove the carboxylic acid, we end up with an amine. All right. So remember those two steps. All right, we'll start with the biosynthesis of catecholamines. We'll start with phenylalanine. Everybody else starts with tyrosine, but that's fine. We're going to start with phenylalanine, and we're going to hydroxylate its R group and make tyrosine, and then another enzyme, a hydroxylase enzyme, will hydroxylate it again. And now we have phenylalanine that's been hydroxylated twice, and that's actually what DOPA stands for. And so that's your trivia question. Find out how many people out there know that DOPA stands for twice oxygenated phenylalanine, dihydroxyphenylalanine. That's what DOPA stands for. And so that's why we started with phenylalanine. We hydroxylated it twice. Now we have L-DOPA. L-DOPA was the miracle drug in Awakenings with Robin Williams and Robert De Niro. Great movie about the tales of Oliver Sacks, a great neurologist. And L-DOPA was the miracle drug. We'll talk about L-DOPA when we talk about Parkinson's disease. L-DOPA is still a mainstay of the treatment of Parkinson's disease. All right, well, now we have L-DOPA. To make dopamine, all we have to do is decarboxylate it, and now we have dopamine. And this is a problem. When we take L-DOPA as a pill, your body will just sit there and decarboxylate it before it even gets in the brain. So we'll use DOPA decarboxylase inhibitors to prevent this step happening in the blood. All right. But again, we've hydroxylated twice and decarboxylated. Now we have dopamine. And then we'll hydroxylate it again to make norepinephrine, and then in methylamine in methyltransferase. I think that's I'm sorry, we have norepinephrine. I'm getting ahead of myself. All right, so we'll reverse that. The dopamine beta hydroxylase will hydroxylate the beta position of the carbon, giving us norepinephrine. Phenylethanolamine and methyltransferase, I don't need you to remember that. But keep in mind, all we're doing is putting a methyl group on that amino, and we end up with epinephrine. So this is a complicated step. Even I can't remember off the top of my head. Phenylethanolamine and methyltransferase is that last step. That's what I don't need you to know. What I want you to know is hydroxylation and decarboxylation. Beta decarboxylation. All right, so we went from phenylalanine to tyrosine to dopa to dopamine, norepinephrine, and epinephrine. These were the enzymes involved: hydroxylase, decarboxylase, hydroxylase, and phenylethanolamine and methyltransferase. All right, and what I want you to know is hydroxylase, decarboxylase, hydroxylase. Right. You need to know about the breakdown enzymes that break down the catecholamines. The end products are HVA and VMA, homovanillic acid and vanillyl mandelic acid. There's all sorts of intermediate steps. If you look at a real pharmacology book, they'll show you all those intermediate steps. But what I want you to know is that this is mediated by two enzymes. Monoamine oxidase and catechol-O-methyltransferase are the two enzymes that break down the catecholamines into their end products, HVA and VMA. And so let's say you have a pheochromocytoma, pheochromo, that color word, because of all that hydroxylating the phenylalanine, those aromatic rings. And so pheochromocytoma is a tumor that makes too much epinephrine. And well, the epinephrine is broken down immediately. You can't measure the epinephrine in the body, but you can collect their urine for 24 hours and look for these right here. And so if we collect somebody's urine for 24 hours looking for the byproducts of the catecholamines, then this can tell us. And so if we have somebody who we think they have a epinephrine, we think they have a pheochromocytoma, they have high blood pressure, they have headaches, they have a fast heart rate, well, then maybe we'll collect their urine for 24 hours and see if they have HVA and VMA as their breakdown products. All right, I'll point out right now that monoamine oxidase also breaks down serotonin, and if we block this step, very dangerous things can occur. So later on in central nervous system pharmacology, we'll talk about monoamine oxidase inhibitors. Monoamine oxidase inhibitors have the most potential for toxic interactions because they break down, blocking monoamine oxidase causes toxic levels of this and serotonin to occur. So we have to be really careful about monoamine oxidase inhibitors. That's why we talk about that. All right, let's talk about serotonin, 5-hydroxytryptamine. We'll start with tryptophan, and we'll hydroxylate it. And now we have 5-hydroxy-L-tryptophan. And then the next step is to decarboxylate it. And we go from 5-hydroxytryptophan to 5-hydroxytryptamine, serotonin. 2 steps: hydroxylation, decarboxylation. That's all I really want you to know about this. All right, we'll go to a monoamine oxidase. We just got through talking about monoamine oxidase breaking down the catecholamines. Monoamine oxidase breaks down serotonin as well. And by oxidizing that single amine, we end up with 5-hydroxyindoleacetic acid. And so maybe we have a type of carcinoid tumor that's making too much serotonin by mistake. And this tumor, we can't just measure serotonin levels, but we can collect their urine for 24 hours and see if they have high levels of 5-HIAA. And if they have high levels of 5-HIAA in their urine, that tells us there's elevated serotonin coming from something, usually carcinoid tumor. All right, so there's 5-hydroxyindoleacetic acid. There's the indole ring. There's the hydroxide in the 5th position, and acetic acid is just vinegar hanging off that 5-hydroxyindole ring. So that's where those words come from. I don't expect you to know any of that. I just wanted to spend some time throwing the words around to show you they actually come from a real place, despite the fact that they expect you to memorize them as if they have no meaning whatsoever. Histamine's pretty simple. We'll start with L-histidine, which is an amino acid, and we'll decarboxylate it. And now we have histamine. The same histamine that mediates allergic reactions is the same histamine we're going to use as a neurotransmitter in the digestive system in the stomach. Right. GABA, again, gamma-aminobutyric acid. We're going to start with glutamic acid. Glutamate is glutamic acid. If you take a acid and you put the 8 word on it, all the glutamate is, this is glutamic acid because this proton's ready to come off. And once that proton comes off, it's glutamate. All right, so glutamate and glutamic acid are the same thing. All right, citrate, citric acid, those are all the same thing. The only difference is the ionic state. All right, so we're going to start with glutamate. We're going to decarboxylate it. And we're going to have gamma amino butyric acid because now the amino group is in the gamma position. All right? And then GABA will be recycled into glutamate, and I don't need you guys to know those steps. Okay, so again, the important steps of that were hydroxylation and then decarboxylation, and we'll talk more about that when we get into central nervous system pharmacology. Well, that's more than enough for neurotransmission. When we come back, we'll talk about 2.2 autonomic nervous system pharmacology, so have your thinking caps on. Until then, aloha.